Okay, this one we're going to talk a whole lot about eyes. So we're going to talk about how eyes can focus light. We're going to talk about the ciliary body and lens and how that helps to focus light. We're going to talk about the different kinds of photoreceptors, rods versus cones. Rods do like light levels, darker light, whereas cones do our color vision. We're going to talk about signal transduction, so how they turn photon impulses into action potentials to our brain. And then we'll talk about receptive fields, which are really helpful for how your body recognizes edges. We'll talk about AMPA receptors versus mGlu-R6 receptors. Off-center, on-center could also be an off-center, and then whatever the center is, the surround is the opposite, but we'll get there. This is a brief drawing of an eye. So say we have some point of light all the way out here, some kind of dot, say it's a box, and that goes here. And whenever you hit something convex, it bends light in. So it's going to bend light just a little bit in, and then it's going to hit the lens, and then it will bend even more. And so the goal is to hit here and then kind of show approximately the same image there. If we were to just do a dot, put a dot here, radiates light in a whole bunch of different ways. Bends light again. And then you get a single dot where that is. And so this helps. This is your cornea. This is filled with what's called aqueous humor. And then in here, it's filled with more of like a gel type fluid called vitreous humor. And so this, your cornea doesn't change, so it will always bend light in the same approximate way. But what happens if we have something that's really, really close? I'm going to use a different color. So you have something like really, really close, and it's sending light here, bends in, and then it bends in further. That ends up being really, really bad. So we don't want this. So what our eye does, I'm going to erase the green for now because you don't need to know that. Well, I just taught you it. Okay. Okay, let's fix up our eye. There's our pupil. One little hole right here. Here's the cornea. That's where the eye can go. Oh, well, where light can go. And then here's our lens. So what happens is, so we have ciliary muscles. If we were to draw kind of like a flat section like this, if we're just looking at it straight on, we have a whole bunch of little ligaments. And then we have the smooth muscle around it. And so as the smooth muscle contracts, these ligaments, the space between them gets shorter, so they go slack. So there's less tension pulling on the disc on your lens. And so that allows your lens to go from more of a straight to a flatter, and it will allow it to bend light more. And that's really helpful for things super far away. So if it's really far away, it's coming in from here, bends a little bit. If it's really, really convex, it will bend the light even more because it's coming from further away, and will allow it to hit there. When it comes to something, oh no, we want it if it's really, really close. Sorry, take that back. If it's really, really close, right here, if we let this bend out more by contracting the ciliary muscle, our lens will our lens will kind of like become more of a bulged teardrop shape, which will allow the light to bend more. Yeah, let me draw. It's kind of like that. and then allow it to bend a little more, showing proper vision. When we relax the ciliary muscle, it extends, and then these little filaments start to stretch. And so that causes the lens to go from this more teardrop shape into a thinner, more flat, kind of like a plate shape. So if I erase that, let's fix up our eye again. There's our pupil, here's our cornea. Now I'll draw a really, really thin disc. Well, thin lens. If it's really, really thin, that helps for far away vision, so we're all the way out here. And this isn't traveling as far. So it's got a less aggressive angle, so it doesn't need to be bent as much in order to hit right here when we're focusing on something. And that area that we're focusing on is called the macula, where you have a dense amount of these cones, which help with color vision. So, if the ciliary muscle contracts, these little filaments become less tight, which means this becomes more teardrop shaped. If this relaxes, so dilates, these become tight, and the lens becomes more like disc plate shaped. And that's how our eyes are able to focus on things like far away, super close. And when you get old, your ciliary muscles become weaker, your disc doesn't become as pliable, and then it becomes harder to see stuff further away or closer, depending on how you've used the muscles. Okay, and we're going to talk about photoreceptors. So, explained, rods do general light, so light versus dark, and then cones do color vision, so specific wavelengths of visible light. So I'm going to draw, these are just our general photoreceptor. And I do this because they've got like a whole bunch of helical parts, gives it lots of surface area. Do I have enough space? Yeah. There's our bipolar cell, and then that's our ganglion cell. Okay, so our photoreceptor, it has a really interesting protein, which is more or less kind of just like closed off. So it doesn't really let anything happen to it, but inside it, it has this thing that's called 11-cis retinol. And so what that means, so it's you've got a fun benzene ring. So it looks kind of like this. And so the cis means these are facing the same way. When light hits this inside here, it turns it into 11-trans-retinol. 11-trans-retinol. And so what that does is it essentially makes it turn into that. So it breaks it off and flips it, more or less. And so when this happens, it causes a bunch of huge cascade of stuff that happens now that I will now go through. So in here we have PDE, so phosphodiesterase, which is really useful for breaking down cyclic AMP or cyclic GMP into its non-cyclic form, so just GMP or AMP. Okay, we have here this is a sodium channel, right here is a voltage gated calcium channel. Here's all of our vesicles. There are snare proteins there. So what happens is normally within the cell, I'm going to use blue for this. So within your photoreceptor, when there's no light, attached to this is cyclic GMP. And so cyclic GMP, is it cyclic GMP? Either cyclic GMP or cyclic AMP. I'm pretty sure it's cyclic GMP. So cyclic GMP naturally is attached to this, which allows it to stay open. And so sodium can come into the cell. When sodium comes into the cell, it depolarizes it, opens this voltage-gated calcium channel, causes us to come in, releases glutamate, which is our neurotransmitter within our eyes. Glutamate. So releases glutamate, and I'll talk about that in a second. When light hits the photoreceptor and hits this protein, it turns the cis-retinol into the trans-retinol. And what that does is that activates, so this is a G protein, activates the alpha subunit, and this turns on the PDE. When the PDE turns on, it converts the cyclic GMP into just regular GMP. Because there's no longer cyclic GMP attached to this, this closes. So sodium no longer comes in here, which means this doesn't depolarize. And so this calcium channel also closes, which means these don't happen and I have less glutamate that is being released. Make sense so far? Great. Our bipolar cells will have one of two different types of receptors. The one that I'm going to talk about right now is what's called mGlu-R6. And so this is a G protein channel. A G protein, I'm going to draw in. This is just a calcium channel. So calcium will naturally be flowing into this cell. This one also has vesicles and will also release glutamate, which is an excitatory neurotransmitter, onto this. So we have light hit this, activates PDE. Cyclic GMP is converted into GMP, closes this off. This voltage-gated calcium channel is then closed, which means I'm not releasing any of this stuff. Because I'm not releasing any glutamate, this mGlu-R6 is not being activated. If I release glutamate from here, which would normally happen if there's no light, it activates mGlu-R6, which when mGlu-R6, the alpha subunit comes up here, binds to this calcium channel, and closes it off. So this closes, which means calcium doesn't come in, which means I'm not sending any signals. And the brain, which means I don't depolarize this, which the brain interprets as darkness. So let's go through if I hit this with light. If I hit this with light, I activate PDE, which breaks down the GMP. Well, breaks down cyclic GMP into just regular GMP, which means my sodium channel closes. If my sodium channel closes, I don't depolarize the photoreceptor, which means this voltage-gated calcium channel doesn't open. Because this doesn't open, I don't release these vesicles filled with glutamate, and I do not activate the mGlu-R6 channel, the mGlu-R6 G protein. So because I don't activate it, my calcium channel over here stays open. So calcium is allowed to run into the bipolar cell and release these vesicles filled with glutamate, which will then activate receptors and depolarize my ganglion cell, which then goes to the brain and says, hey, we're seeing light. Now, the other important one is instead of mGlu-R6, some have an AMPA receptor. And if you remember, AMPA receptors are ion channels. So if I am in dark, this does not activate PDE. If PDE is not active, GMP stays as cyclic GMP, which means this sodium channel is open. Sodium comes in, depolarizes the photoreceptor, which causes this voltage-gated calcium channel to open, release of glutamate. When glutamate binds to this inotropic receptor, it allows cations to flow in and depolarizes this cell. This causes the release of the glutamate, which tells the brain, hey, I'm seeing light. So all photoreceptors will do this. What determines whether or not the brain says I'm seeing light or I'm not seeing light is what kind of receptor is on the bipolar cell. And so this is where on-center and off-surround are really important. Okay. So I'm going to draw it this way. This is our ganglia. This is our bipolar cell, and then these are our photoreceptors. And so I'm going to stick on this cell an mGlu-R6 receptor. So this is what's called an on-center, off-surround. So on, and then these two are off. And if you were to look at the back of the eye, say I'm looking at inside the retina, you would end up finding tons of these little receptive fields. As you get further away from where the macula is, you get larger receptive fields. When you get closer to the macula, you get smaller receptive fields, even to the point where the center is literally just a single photoreceptor. Okay, so here, this is our on-center, off-surround. And I'll draw a little. Here's our center, which is on, and then we've got off. Okay. So, if I were to have light, I'm going to use, let's say this red. So I've got some light that's shining on just this area. So what happens here? So this is in the dark. Because this is in the dark, that means cyclic GMP stays bound to that sodium channel, which means I have calcium coming in, this depolarizes. This sends glutamate. Send glutamate. This activates the mGlu-R6, which deactivates my calcium channel here, so this does not depolarize. Now the interesting thing is for these ones, we have the same thing. This ends up releasing glutamate because all photoreceptors in light will release glutamate no matter what. The cool thing is what we have here. So those little red boxes are what's called horizontal cells. And so they have, when they interact with glutamate, they're depolarized, and they end up releasing a neuromodulator called GABA. And so GABA is your main inhibitory neurotransmitter. And so what it does is it will end up opening either chloride channels or potassium channels, which ends up causing this cell to hyperpolarize, which means it releases less glutamate. If I have less glutamate, mGlu-R6 is activated less, which means my calcium channels are closed less. So I still have calcium coming in, which means I end up releasing glutamate here. And then this ganglia tells the brain, I'm seeing some amount of light. Okay, so now you're gonna be like, oh no, you said that this could be on or off. That's right. Okay, so we're gonna switch these. What if this is now an on surround and an off center? So we'll change them up here. And so the interesting thing is for off center, on surround, you do not have mGlu-R6. You have that AMPA receptor. So in this example where I have light shining only right here, if I have light on this on portion, light here causes less release of glutamate, which means this cell isn't depolarized as often, which means I'm releasing less GABA, which means this cell is allowed to depolarize more often, which means it will end up releasing more glutamate. Because it is in the dark, no light means this cell ends up depolarizing because sodium channels open, calcium channels open, releases vesicles, releases lots of glutamate. And because it's not being inhibited by this one, it's allowed to release more. However, on the other side, this is in the dark. This is dark, this releases glutamate, actually, which causes this to activate, releases GABA. And this causes this one to depolarize slightly less often, so you don't release as much glutamate as you possibly could. So this GABA, not that, this glutamate binds to the AMPA receptor and allows sodium and calcium to run in, which depolarizes the bipolar cell, which then sends glutamate into the ganglion cell and tells the brain, I'm seeing light. And so this is really important. We're going to draw a couple examples. Okay, so we're going to draw some circles. And so this is really important for edge detection. See those? Perfect. Okay. These are all going to be the same thing. These are all off center, on surround. And then I'll have a second set over here, which is going to be our on center. On center, off surround. Okay. So if I've got light here, say we've got a beam of light slowly passing over. So if you remember as we went through, I'm going to draw kind of like the frequency of action potentials. So I have light here because this is on, I'm causing the less glutamate to be sent, which means less GABA, which means my ganglion, my main photoreceptor, gets to send more glutamate. Yeah. Which ends up depolarizing the bipolar cell, and it says, aha, there's light here. So for this one, we're going to kind of just put it in your centers are stronger in sending action potentials than the surrounds are. So if we give it, say this whole receptive field can end up sending a maximum of say 10 action potentials in some amount of time. This one can send 2, this one can send 2, and this one can send 6. So I have light here, and the on or off tells you what happens to the bipolar cell. So if this is on, and I'm hitting light, and light is hitting it, that means it's going to activate the bipolar cell. If it's off, and there's no light, it's going to activate the bipolar cell. So here I've got 2, and because there's hitting light, I am going to send those 2. This one, because it's an off center and there's no light hitting it, I'm going to send it 6. My on center here, my on surround, it's dark, it's an on, it's not sending its stuff and trying to prevent action potentials being sent. So this ends up sending a total of like 8 action potentials. 5, 6, 7, 8. So high frequency. Now, the light is traveling here, and now it's covering both of these areas. So now we're going to do same thing. So we've got our 2, 6, 2. Actually, I'm going to write these for all of these ones. And we'll do them over here as well. Okay. So, let us continue. Light hits here. This is an on. It likes that. It depolarizes the bipolar cell. Light is hitting the off center. It doesn't like that, which means it's not sending its action potentials. This one is in the dark. It's also not sending its action potentials. So this is only getting like 2 action potentials every some amount of unit of time. So not super frequent. In this one, we're going to have light hit the whole thing. Okay, so as before, this has light, it likes it. Light is hitting the center, doesn't like it. Light hitting here likes it. So this is kind of a semi-more frequent one. And then we can continue. And then, okay, light has moved past. That was a bad line. Light has moved past, and now light's here. Okay, so this doesn't have light on it. It's not being activated. This has light on it, but it's an off center, so it doesn't like that. And then this one is also being sent, which means we only end up getting 2 action potentials. And so you can see when you've got this off center on surround, as you have something pass over, or like you detect some kind of edge, you have a high amount of action potentials that goes low and then goes low. Yeah. So you've got here, you've got some kind of edge here, and it's sending a lot of signals saying, hey, we've got something really bright over here, but something really dark over here. And that's how you can detect those edges. And so if we do the same thing for this side, I'm going to use red again to describe the light. So we've got light here, and then we've got light here, light on the whole thing, and then light on just that part. And we can go through, do the same idea. Light here, doesn't like it. Light here, doesn't like it. Light here, yes. So we've only got a couple action potentials every so often. Then what happens with this one, we've got light here, but it doesn't like that one. We've got light here, but it does like that. And light here, and it does like this one. So we've got a lot of action potentials. That's 7, 8. So high frequency of action potentials. Here, we've got light here, and it likes that, but the 2 sides don't like that. So we end up only getting 6. Okay. Ooh. For this one, we've got light here, likes it, dark here, and because it's an off surround, it appreciates that, anamorphically. And so we've got high action potentials again. Lots of action potentials. And then, yeah. And so you can see with the on center, off surround, as you increase the amount of light, like as you're passing it over, you get high amounts of action potentials, and then it starts getting lower. And so again, this is also really helpful for edge detection. And that's receptive fields. And so your AMPA and mGluR are what determine whether or not it's an off center or an on center, and then vice versa for the surround. If it is an on center, that means you have an mGluR6 receptor on your bipolar cell. If it's an off center, that means you have an AMPA receptor on your bipolar cell. And that's light and eyes.